Uh, well, I think it's afternoon now, so good afternoon, everyone. I would like to start by thanking the organizers for allowing me to present remotely. Unfortunately, due to bureaucracy, I couldn't make it in person to Vegas. My name is Oriol. I'm the deputy group leader of the Wasserman Lab at UBC, and today I will present ExplaiNN, our approach to explainable artificial intelligence that I have co-developed with German and Manu. By the way, Manu is attending the conference in person, so feel free to reach out to him after this presentation if you want to discuss ExplaiNN in more detail. I'm sure that it will be his pleasure. Sorry, Manu. Sequence-based deep learning models, and in particular convolutional neural networks, have shown superior performance in a wide range of genomic tasks, from predicting chromatin accessibility to transcription factor binding sites. A limitation of these models, however, is that they are difficult to interpret, which has slowed their broad adoption. For example, what features has the model learned and how do they influence the predictions it makes? Being able to interpret the model in this way, that is, knowing what it has learned, it's just as important as having a good model that makes accurate predictions. In genomics, where the input data to deep learning models are typically DNA sequences, there are many different approaches to interpret the model. For instance, we can interrogate the model to find out which DNA motif a particular node in the network has learned, or what is the importance of each nucleotide in each input sequence according to the gradient of the model. Moreover, if the model was able to learn nonlinear interactions, for example, through the use of fully connected layers, we can interrogate or we can ask what are the relationships between each pair of motifs that the model has learned. If you wish to learn more about model interpretability in genomics, I highly recommend this review from our lab and the Liebrecht and Mostafavi labs. ExplaiNN, which is the topic of my presentation today, stands for Explainable Neural Networks and is an adaptation of neural additive models for genomic tasks. The architecture of ExplaiNN consists of multiple independent convolutional neural networks that we call units. Each of these units, highlighted here, contains one convolutional layer with one filter, and this is followed by an exponential activation function and fully connected layers. Then, in the final linear layer here, the model combines the outputs of all these units as in linear models to compute the predictions. I want to highlight that the architecture of ExplaiNN is very flexible. For example, we can specify what's the length of the input sequences that we want in the model, what's the number of these units that the model is going to have, what's the size of the convolutional filters that the model has or each of these units has, and whether or not we want to use fully connected layers and how many. ExplaiNN provides different levels of interpretation. For example, we can derive DNA motifs from the convolutional filter of each unit as follows. First, for each filter, we can scan both strands of well-predicted sequences. And by well-predicted sequences, I mean positive sequences that the model predicts as ones and negative sequences that the model predicts as zeros. Then we can derive a motif by aligning all k-mers present in those sequences that activated the filter by 50% or more of its maximum activation value. And next, we can assign a biological annotation to the filters unit by comparing the motif that was derived from that filter to motifs from the JASPAR database using tools like TomTom. In this particular example, since the motif looks like the motif of a GATA transcription factor, we can refer to that filters unit as being GATA-like, since it most likely helps detect the presence of GATA motifs in the input sequences. Also, as in linear models, we can use the weights of each unit from the final linear layer for interpretation purposes, as they often relate to how much each unit contributes to the prediction of each task. Finally, for each input sequence and for each unit, we can multiply the output in that unit by the unit's weight from the final linear layer. We refer to this product as the unit importance scores, and it reflects whether the unit is relevant to the prediction of the sequence. As proof of concept, we first applied ExplaiNN to predict the binding of 50 transcription factors to open chromatin regions from a reference dataset. In ExplaiNN, one of the most important hyperparameters, and as you can probably guess already, is the number of units in the model. To assess the impact of the number of units on model performance, we trained several ExplaiNN models on the previous dataset using an increasing number of units, from 1 to 200. As expected, the performance of ExplaiNN, shown in green, increased with the number of units used, as measured by the average area under the precision recall curve. For comparison, we evaluated 2 additional models on the same dataset: a deep convolutional network with 3 convolutional layers and 2 fully connected layers that we call deep CNN, and DanQ, which is a hybrid deep learning model with convolutional and recurrent layers. Both of these models have more trainable parameters than ExplaiNN, making them more complex. And as you can see, ExplaiNN outperformed the deep CNN model, and when using more than 100 units, it nearly reached the performance of DanQ. Next, for each of the ExplaiNN models that we trained before, we derived a motif from the filter of each unit. And as you can see, it happened the same as with performance. The number of non-redundant motifs that the model was able to learn, indicated on the y-axis, also increased with the number of units used. Also, the number of non-redundant motifs derived with the ExplaiNN model trained using 100 units was similar to that of DanQ when using TF-Modisco clustering on deep lift attribution scores, both for motifs of the free-state transcription factors, shown in green, and for motifs of other transcription factors, shown in gray. Combining features of linear models, one of the advantages of ExplaiNN is that we can interpret the contribution of each unit to the prediction of each class by visualizing the weights from the final linear layer. For example, as you can see here, several units with filters that were annotated as CTCF motifs or CTCF-like had high positive weights for predicting the CTCF class, indicated with black arrows. Similarly, 2 Fox-like units had high positive weights associated with predicting the FOXA1 class. An alternative way that we have to interpret the contribution of each unit to the prediction of each class is by calculating unit importance scores. Shown in the right, you can obtain them by multiplying the output and the weight of each unit for each input sequence. Here, we took one of the previous FOXA1 units, Fox-like units, and visualized the importance scores. And as you can see, this unit was important for predicting the FOXA1 and the androgen receptor classes, indicated with red arrows. And this is in agreement with the literature. For comparison, we took the DanQ model and we nullified the Fox-like filters in that model one at a time and calculated the impact of the nullification process on the model outputs. In other words, we set the activation values of the filter across all sequences to 0 and computed the impact on the model's predictive capacity. And as you can see, the impact scores from the nullification process were consistent with unit importance scores. So after exploring the performance capabilities of ExplaiNN and the interpretation capabilities of ExplaiNN and the transcription factor binding data, we turned to a more challenging task, such as predicting chromatin accessibility in the mouse immune system. In the original work, the authors used a deep neural network called AATAC to analyze and interpret the data. And here, we trained ExplaiNN on the same dataset and checked whether it could recapitulate the same findings as AATAC, which is a more complex model as well. In the original work, the performance of AATAC was reported as the Pearson correlation coefficient between predicted and actual accessibility values, indicated here on the y-axis. And as you can see, the performance of ExplaiNN saturated at around 300 units and nearly reached the performance of AATAC, here shown in gray. Moreover, the predictions of the 2 models were highly correlated. Next, we annotated each unit and visualized the weights from the final linear layer of the ExplaiNN model trained using 300 units. And doing so revealed different lineage-specific markers, which are here highlighted in black. For example, as you can see, a CBP-like unit that had high positive weights in myeloid cells was also very important in predicting accessibility in those cells, in agreement with the role of these transcription factors in regulating myeloid differentiation. The same was true for a PAX5 like unit and B cells, which is consistent with the role of PAX5 in establishing B cell identity and function. One limitation of using DNA motifs as the basis for interpretation is that we cannot distinguish between transcription factors from the same family, as they typically have very similar motifs. For example, when the filter of a unit looks like an AP1 motif, we cannot tell whether that unit helps predict FOS and JUN, or BATF and JUN-B, or a different AP1 heterodimer. Because like you can see, all members from the AP1 family, the motifs of all these transcription factors, they look very similar. So to obtain units that could provide such a level of resolution, that is, that could distinguish between members of the same family of transcription factors, we implemented a transfer learning strategy. First, we pre-trained 350 single-task DANQ models, each predicting the binding of one transcription factor from the REAM database to the mouse genome. And next, we initialized an ExplaiNN model with 350 units and replaced each unit with one of the DANQ models that had been already pre-trained. We then fine-tuned the ExplaiNN model on the A-attack dataset, freezing the DANQ models. And by freezing, I mean that the weights of the DANQ models were not allowed to be updated during the fine-tuning process. They remained exactly the same. Next, we visualized the importance of each DANQ model unit, which allowed us to highlight the importance of individual transcription factor family members. And in this particular example, we see units corresponding to the IRF4 and IRF8 DANQ models, which were important for predicting accessibility in different immune lineages, all of them in agreement with their distinct roles in regulating the immune system. Similarly, the unit corresponding to the PAX5 DANQ model at the top was important for predicting accessibility in B cells, which is consistent with the role of this transcription factor. However, the units corresponding to the PAX3 and PAX7 DANQ models at the bottom were not relevant for any immune cell lineage, which is consistent with the role in regulating myogenesis and not the immune system. And these are the conclusions of my talk today. First, that ExplaiNN is an explainable deep learning approach designed for the discovery of genomic features contributing to model performance for a wide range of predictive tasks related to DNA sequence data. Furthermore, that ExplaiNN nearly reaches the performance of DANQ and A-attack in predicting transcription factor binding and chromatin accessibility. Regarding interpretability and in terms of motifs, that ExplaiNN provides similar results than DeepLift and TF Modisco. And besides, that unit importance scores correlate well with impact scores from filter nullification. Finally, that when combined with transcription factor binding models, ExplaiNN can highlight the importance of different paralogs. If you liked ExplaiNN and if you wish to find out more about it, here are two links to a bioRxiv preprint and a GitHub repository. In the GitHub repository, you will find several Jupyter notebooks that will help you replicate the results shown in the preprint, as well as a series of Python scripts that I have to push yet in the next days to the beta branch that will facilitate the training and interpretation of your own ExplaiNN models. I would like to end my presentation by thanking the support of the members from my lab and our funding agencies. And now I will be taking your questions.
With these more complex models, whether the additional training time actually gives you a huge boost in performance. So it's really nice to see a simpler model that's able to do similarly. A question I had in terms of the interpretation is you showed some really nice results showing how your neural network is capturing known biology. But we know that if we use the known biology as features, our models don't do nearly as well as these neural network models. So I was wondering, what have you found when interpreting that is not something you already knew? And is that something you've been able to, learn more about or follow up on?
Yeah, so that's a very good question. And I am just going to comment on, unpublished work. So we, for example, used ExplaiNN in order to find similarities and differences between the human and mouse genomes that regulate the human and mouse genomes. And on the one hand, you find things that are obvious, such as G quadruplexes, preferential CTCF binding in mouse, but then you find things that are very different, such as, of course, also, species-specific repeats, but you also have differences such as motifs that we don't know what they are. We think they are zinc finger motifs because they look like motifs of zinc fingers, and actually those motifs that we have no idea what they are, they were actually important for distinguishing sequences that were either human or that were mouse.
Thank you. Love to learn more about that.